All right, so first thing I want to point out, anybody who's ever seen me talk before, I talk very fast, and I'm going to give you way too much information. And so this will be available as a PDF, and it's being videotaped, and you'll be able to find these and have it go by really fast again. So, and if there's anything you find interesting or questions that come up, I always tell patients, you may email me. And I get emails probably twice a week from all over the world, from patients that have either seen videos or just things, and I will answer eventually every single one of them to the best of my ability. Because I've learned, the reason I'm here is because I actually learn a lot from you all, actually, and you'll get a feel for that as I go through this talk. Other most important thing, I'm only part of the story. Back there in the back is my wife, Takako Jones. We are a husband-wife team. We are truly co-PIs. We work together. We have one car, we go, we come, we work together, and she does basically all the work, and I get all the credit. So we're going to give her the credit this time. All right. I just want to disclose, just to be clear, because I'm going to be talking about some therapies. You know, we do have some patent applications, just to be clear on conflict, and I have a scientific advisory board for a pharmaceutical company. And sometimes people see this and they think, oh my God, you know, pharma. Well, pharma is great, because if pharma wasn't interested in FSHD, we'd never get anywhere, okay? And a few years ago, pharma wasn't interested. So this is actually a huge advancement and a huge plus, okay? So just to be clear, but just as a disclosure. All right, so sometimes in FSHD, you may actually feel like you're kind of maybe a little bit lonely, but actually, you know, there are neuromuscular diseases, about 30 muscular dystrophies out there, and of which FSHD is one of the most prominent ones. And what I get emails, not just about FSHD, but anytime something's in the news for any muscular dystrophy, the patients email me and say, what does this mean for FSHD? And normally it means a lot, because advances across the neuromuscular disease fields actually oftentimes can be applicable to FSHD. Therapeutic advances, things that we understand about disease progression, maybe directly, maybe indirectly. Viral delivery of therapies, small molecules, the types of things that Amanda was just talking about. There are a lot of, this is myostatin inhibitors. There are a lot of things that are applicable across the disease. So you're not alone. There's a lot going on. And everyone in here that's over 35 has a muscle disease. Okay, about age 35 on, you are fighting a muscle disease, okay? Aging is, okay? So this is what we tell Congress, right? And we're gonna tell NIH, fund muscle diseases, okay? Because you got it. So the way MDA puts this, research across the full spectrum of neuromuscular diseases leads to medical breakthroughs that accelerate treatments and cures, right? Curing SMA is great for FSHD. Okay, we need successes across the board are good. They're going to increase funding, they're going to increase development, they're going to increase pharma interest, and they could be directly applicable to FSHD. Okay, disease progress brings urgently needed answers to affected patients and families. So, I'm going to step back a little bit. I am not a muscle person. Okay, I was not trained in muscle, I didn't know anything about muscle. My lab is an epigenetics lab and developmental biology. These are worms, C. elegans. We do some DNA methylation analysis, which you've heard a little bit about. Yeah, here's a mouse embryo. My wife's stained. Here's a Drosophila. We worked on all sorts of developmental biology questions. Okay, nothing about muscle. I started my lab at the University of Illinois in 2001. 2002, this kid went to one of these meetings and was sitting out in the audience and heard someone say FSHD is an epigenetic disease. Now, why would he be out there? Because he had just been diagnosed his junior year of college as having FSHD, and he wanted to learn about it. He happened to be a student at the University of Illinois, where I happen to have my lab that works on epigenetics. So this kid came, Ryan Wibbles, came to my lab and said, can I join your lab and work on epigenetics of FSHD? I said, sure, because that's what junior faculty do. We don't know any better. So sorry, it's true. I mean, we didn't know what we were doing. Thankfully, the FSH Society knew what they were doing, and I got to learn a lot about it. They helped get us going. So he introduced us. So a patient sitting out. So this gets to kind of what Mark was talking about earlier. You can make a difference, right? This is one person starting. Now, my lab is 100% full-time. We're one of the best funded NIH-funded labs in the country on FSHD. We've produced other researchers. We have pharma connections. We developed, all because one person sitting in the audience didn't, you know, we say just because you can't stand up, you don't have to take it sitting down, you know, okay? You can do something about it. You're not alone. Okay, so what is epigenetics? This is kind of, you hear DNA methylation? I'm an epigenetics person. I teach a class called epigenetics. It meets 45 classes to get you a little bit of understanding of epigenetics. And you're going to get it all in just really quick. So, non-Mendelian pattern of heredity, context dependent, influenced by the environment. What this means is that your DNA sequence can have the same sequence, but the environment inside a cell is different. And there's a great example. These mice are genetically identical. They are epigenetically different. The context of the certain genes in their genomes are different. Calico cats, you know why calico cats are always female? Because the genes, these are on the X chromosome, and you have epigenetic regulation of the X chromosome. In males, you only have one X, and female, you have two. So calico cats are always female. These are the Drosophila eye. I'm going to show you in a second. You have a white eye cell, you have a red eye cell, genetically identical, epigenetically different. The context of a genetic sequence matters. And this is key because this context can be influenced by the environment. It can be influenced by the environment in vitro, in utero, right? What mom eats matters. Okay, it can be influenced by the environment that you live in. Okay, genetics don't change so much. Epigenetics can change. All right, here's just an example, kind of a textbook example. Here's a Drosophila. You always say fruit fly research, stupidest thing ever. Not really. We've learned a lot from these model organisms. Drosophila fruit flies normally have a red eye. If you mutate the white gene, they have a white eye. This is a genetic mutation. All the cells have white. Here's an epigenetic mutation. You take the white gene that is in a nice active part of the genome, and if you just, and you move it, the exact same gene sequence to a region that is epigenetically off, the gene is off. The exact same sequence in a different environment is expressed or not expressed. This is what we mean by epigenetics, and in people, this is actually regulated by DNA methylation. Here's our mice, again. These mice are genetically identical. The difference between these mice, we can do this in mice, you can do this in some probably human populations, we don't like that. But in mice, you want to do it. And these are influenced, what you fed mom while they were in utero changed their epigenetics. Genetics are the same, epigenetics are different. And you don't just have a brown mouse or a yellow mouse. Actually, these yellow mice are going to spontaneously develop tumors, okay? They're going to be obese, okay? Genetically the same, fed the same, controlled in the lab. You change the environment of this one gene, and you got a mouse that's going to develop tumors versus a genetically identical one that's not. This is epigenetics. This is DNA methylation, the effect of this that can have, okay? So, affects long-term health. This is very important. So why do I care? Genetics of FSHD are really complicated, as we heard this morning. Epigenetics are extremely simple. Bottom line, if the FSHD region is in an what the FSHD mutations do, whether it's FSHD 1, 2, or 3, what those mutations do is they take a region that is normally epigenetically off in the healthy individuals, and the environment changes into an epigenetically on, okay? And when we depict DNA methylation, black here shows that it's methylated, yellow shows the sequence is unmethylated. These are the exact same DNA sequences, methylated versus unmethylated, and you have FSHD or you don't. It's epigenetic. And you can get there either through your deletion, this is your FSHD1, or through FSHD2 without a deletion, which is the mutations in your DNA methylation machinery. Now the cool thing is nature has already cured FSHD. Okay? Talking about penetrance. 1 out of 6 patients that came into our Wellstone clinic as the unaffected healthy control had the exact same mutation as their affected relative. Okay? You can be asymptomatic. You can be healthy and have be genetically FSHD, but epigenetically healthy. Okay? So, therapy. What do you want to do? We just need to, now we don't need to do gene therapy. We just need to figure out how to take this and go to here. And this isn't all the way back to healthy. We just need to go back a little bit, it turns out. This is another thing that my wife figured out. You don't have to dial it back from, it's not 0 to 10. You only got to dial it back a little bit to get from an affected to an asymptomatic. So, since 2003, Ryan was exactly right. FSHD is caused by a loss of epigenetic regulation that he heard someone else talk about. And since 2003, we have been 100% FSHD lab. Okay? And because of and we're studying the epigenetics of FSHD. All right, so 2003 was a rough time to be getting into FSHD. On one hand, if you don't know anything, there's a lot you can do. On the other hand, if you don't know anything, it's pretty hard to do stuff. FSHD gene, didn't know. And one reason I'm bringing this up is because you sit down and the patients, it's never fast enough. Think about this is what in my career has gone on. No FSHD gene, no mechanism. The cellular models were wrong. No animal models. The treatments were, let's just treat it the way we treat any other disease. It's not a dumb idea. There's nothing wrong with it, but there's nothing else to go on. But probably not the best idea, okay? But what are you going to do? Now we are in 2017, okay? We know the gene. We know the mechanism, epigenetic dysregulation that leads to DUX4 expression. Cellular models. You just heard about some great cellular models from Amanda. There's many other cellular models out there. And there are now mouse models, fly models, zebrafish models, because whether or not that's, depending on your therapeutic approach, you may want to have an animal model that's not walking too well. You give it the drug, and maybe it's walking pretty good. Okay, treatments. Now we have myostatin inhibition. We got immune suppression. These are already FDA approved and biology based, and there's a lot more coming, and that's what I'm going to be talking about. All right, so this is the simplified version of what we know. I didn't want to overwhelm you, okay? So this is what we got. DUX4, you've been hearing about DUX4. And the reason I put this up here is, June pointed out we have a target. Well, you also got 10,000 drugs that fail, and you got all these big, little funnels of stuff. I want lots of targets. Okay, more targets is better. More approaches is better. Let's attack this thing from every way you can. So DUX4, great. This is the central player, okay? What do we got? We have antisense technology, morpholinos, PMOs, microRNAs. You don't need to know what these all are. All you need to know is these are different chemistries that are going to wipe out the DUX4 full-length mRNA. They've proven effective in other neuromuscular diseases. This is a case where chemistry and technology for delivery in other neuromuscular diseases is going to be directly applicable to FSHD. Okay? Inactivate or destroy the DUX4 full-length mRNA. And the only one that's publicly out there on the website, even though there's many more doing this, is IDERA. I'm not putting anything that's out there that's not publicly available. So what's the other thing you can do? Well, let's say maybe let's stop DUX4 from ever being expressed. Instead of having to wipe out the toxic molecule, let's never make the toxic molecule. Okay? We know now how DUX4 is regulated, epigenetically and also through environmental factors, through some other regulatory mechanisms, some hormone signaling pathways, some myogenic factors. These are all targets to prevent this from ever even getting, instead of, how to wipe out the bomb, let's just never let the bomb get there. Okay, so small molecule inhibitors, CRISPR technology, which I'm going to talk about, prevent the expression, Fulcrum, and Janae, this is some of what, again, like Amanda was talking about, these approaches to kind of prevent the expression. All right, now we're talking downstream, small molecule inhibitors. You can block, so let's say that's all well and good. Maybe those don't work. But we know what happens when DUX4 is expressed. You get immune response, you get some aberrant expression genes, you get some sort of cell death, you screw up muscle development, and a bunch of stuff we probably don't know yet. These are all targets, because now you can just maybe block the bad stuff. So you look at everything up here that you should see, you say this is a therapeutic target. And that's good, because you know what, there are some FDA-approved molecules that can actually affect some of these things. There's a lot of work to be done, but we're not stuck with one target, okay? Then you have DUX4-independent approaches. Again, this would be the myostatin inhibition type of thing. Essentially, you can build up the muscle as it's, muscle's being broken down, build it back up, okay? And what you're probably going to end up with, it's combinations of this, right? Combinations. Because let's stop it from going away. Let's turn off the toxic gene, and let's build up the muscle. These types of combinatorial approaches. So there's a lot going on. So what do we work on? So I'm at the University of Nevada, Reno School of Medicine. I just moved there from UMass Medical School. And I'm just going to briefly show you a little bit of what we're doing on small molecules for the epigenetic effectors. We work up here. I'm an epigenetics person. There's several other reasons. And the other thing is CRISPR technology, which you may have heard of in the news all the time. This is genome editing, and we've shown that genome editing is applicable to FSHD. All right, so the question is, can we therapeutically return an FSHD to an FSHD non-effective state? Okay, essentially, can we go this direction? We're not doing gene therapy. We're just saying, can we take the epigenetic status that gives you FSHD, and I'd be happy with asymptomatic. You know, you don't need to be healthy. I mean, you just want to be not nearly as severe. And can we just dial this back? Okay? So this is the simplified cartoon of what's going on at the FSHD locus. And all you need to know is two things. This looks very different than this. And these are all therapeutic targets. Okay? Because this is all this machinery here. This is SMCHD1, the FSHD2 gene. All of this machinery keeps DUX4 off in healthy individuals. When you lose this machinery for one reason or another, this active, the on machines come on. So one thing that you can do, if we want to go from FSHD back, is basically, can we block some of this on from happening? Can we epigenetically flip the switch, right? So basically, all these are targets. Can we go in with known drugs or known approaches that will affect these specific on switches and turn the DUX4 region off? Okay, and so we have shown actually in the lab just briefly. I don't want to show you too much raw data. All you need to know is one of those things on that chart. If we knock it down, DUX4 levels go all the way down, and we don't affect its ability to be a muscle cell, as kind of has been pointed out. Basically, this shows that you can, and we've done larger sequence analysis as well, you can actually knock out some of this and drive an FSHD cell back to a healthy cell with small molecule approaches. That's really good news, okay? Now, of course, there's a lot more to be done. All right, so the second thing, can we therapeutically return an unaffected epigenetic state by recruiting off machinery? So that's a different answer, right? That's a different question. We're going to, instead of killing the on, let's bring in some off. Okay, so we're doing the same thing. Going back the same way, we're just taking a different approach. More method, more target, more is better as far as I'm concerned, because I'm just not smart enough to tell you which one's going to work. So what we have is essentially this off switch, healthy asymmetric. Can we bring in a giant big piece of off and shut this thing down, okay? And that sounds stupid, but that's what the technology basically does, okay? We're going to do this using CRISPR. Okay, and you've heard about CRISPR as genome editing that is going to destroy the world. The next thing that's going to destroy the world, or whatever. It's, part of what I want to do is some advocacy to tell you that's not the whole story. It's actually not as bad as it sounds. It's not as easy as it sounds. All CRISPR is a technology that says you can target the genome. I want to take this whatever to this sequence of the genome. I want to take a piece of on to this piece of the genome. I want to take scissors and cut the genome. All it is sequence-specific genome targeting. You got 3 billion. You got 3 times 10 to the 9th, base pairs, and you're gonna. How do you find the gene that you're interested in? Well, CRISPR lets you do that. You can cut the DNA. That's editing. That's hard. That's really inefficient. I'm not a fan of it. But you can target an activator. You actually can say, I want to turn this piece of the genome on. You can target a repressor. I want to turn this piece of the genome off. Sound familiar? You could say, I want to look at this. There's all sorts of things you can do with CRISPR. It's really a spectacular technology that came out of basic research, actually, which is why NIH should continue to fund basic research since we're going to be on YouTube. So Dr. Himeda in my lab basically asked this question: can she use CRISPR technology as a therapy for FSHD? And so, there's an editing approach, which turned out to be really hard. Basically, could we just cut this DNA up? Well, your genome doesn't like to be cut up. Okay, think about that. That's not a great thing. But could she actually use CRISPR to bring in an off switch to the DUX4 gene? That was basically the question. And so what she did is she basically looked at the DUX4 gene, found very specific sequences within the DUX4 gene, and then took this Cas protein that does not cut, it's called dCas, fused to a transcriptional off complex, and said, can I shut down the FSHD region, can I shut down DUX4 using this technology without cutting it? And the answer was absolutely, she could do that. It turned out this was the first use of this technology, and it scared us to death because everybody started calling us. And we're not a CRISPR lab either. We're not a muscle lab, and we're not a CRISPR lab. So we did the first CRISPR therapy in muscle. So how do you end up doing that? Well, you end up doing that because, and no one in their right mind would have funded us except some disease foundations. And one of the values of funding from disease foundations is they let you do these kind of crackpot, high-risk, high-reward experiments. Okay? And it's really important because these are where the leaps forward. And so basically in a matter of 4 months of never having used CRISPR before, we were able to show that this is a therapeutic, a new avenue, a cutting-edge technology that's going on, worldwide technology for almost every disease out there is applicable to FSHD. Okay? And we never would have been able to get this funded. She ended up winning the FSH Society Young Investigator of the Year award for this work. Okay, so she's fantastic. So so if you're ever going to start a lab, the key is to find brilliant people, and I got 2 of them. Always get people smarter than you. All right, so this sounds right. Now you're going to say, okay, this isn't a lab. We've cured a lot of things in lab, but is this viable? Well, there's a lot of companies out there that are putting a lot of money in to say yes. We think we can get this to clinic, not just FSH, but other. And the thing is, again, this is a case of advances across biomedical research are applicable to FSHD. FSHD, we're not a CRISPR, we're not doing any CRISPR technology. We're using the brilliant approaches, the genius, we're really just hijacking the genius of these other diseases and these brilliant CRISPR scientists and just applying it to FSHD. Okay, so it's fantastic. This is a $5.5 billion industry. There's money to be made. I mean, someone's going to figure it out, right? And that's good news for FSHD, right? Who cares, right? Let's cure it with CRISPR. So, and also it's getting easier and more moving faster to clinic. So the problem is we did that on muscle cells in a lab dish, which, well, look around, okay? Very different. How are you going to do this in a person? Well, FSHD obviously is skeletal muscle disease. So we need a model. We need an animal model. And as you heard, the DUX4 region, the FSHD region, is not present in mice. But that wouldn't stop my genius wife from still making a mouse that gets FSHD. We took the human gene and put it into the mouse. Now, it took 6 years. It was a lot harder than it sounds. I mean, this is simple, right? All we had to do was this. She can explain it. It works great. Failed 3 times. Here's another case we failed. This is like generation 4 or 5 of the mouse. You learn something each time. There's a lot of failure in science, but if you do it right, you control it right, you learn something. And you always are moving ahead. So we moved ahead, and so what do we end up with? We end up with this little guy right here. Okay? This is a mouse that we induced to get FSHD. And there's his siblings that do not get FSHD. And this some of y'all may be able to relate to this little guy. Everyone's running around like crazy around him, and he's just having trouble getting around the cage. Right? So what do you want to think therapeutically? I'd sure like to be able to now inject this guy with something and see him walking just like everyone else. Right? That would be a really good thing. So now we have an animal model. So now we have a mouse model of FSHD, and actually we got several different mouse models for different therapies that we can use. So how are you going to assay it? Well, here's this guy up here. Well, this is the simplest assay there is. You put him on a grid, you hold it upside down. What's going to happen with a mouse? Well, we could, we want to power the, we could hold them upside down for 10 minutes if we wanted to. But you're going to look at these guys down here. These are the mice that we induced to have FSHD. And actually, one of these guys is the same mouse before and after FSHD induction. And what you're going to see is evidently really slow motion. Wow, that's slow. I guess my computer's a little locked up. He's on a pillow. He's landing on a really soft pillow, and he didn't complain at all. Okay? But what it is, it's such a measurable outcome. It's strength. It's grip strength. This mouse is going to hold on if he can. So now I don't need him to for 10 minutes. Maybe if he only holds on for 10 seconds. That's a measurable outcome for a therapeutic treatment. Okay, in an animal, not in a dish now, in an actual animal, right? That actually makes you maybe feel a little bit better about your therapy. So we took that and we put it in, took our CRISPR, put it into a virus, and put it into the mice. And what we found is we could actually knock down DUX4 with a whole body injection into mouse. Now, I don't have a picture of the mouse walking any better. This was just the start of our experiment, but we were able to AAV delivery. So this is actually how you deliver gene therapy to people, and it's starting to work. So when you think about things moving fast, okay, things are progressing. And technology, 2 years ago, CRISPR technology for CRISPR wasn't even on the map. 5 years ago, it wasn't even in the world. So, things are moving a lot faster than you may realize. And now we actually have this project funded by the FSH Society. Okay? Thank you. Appreciate it. And what we're doing, because it's still a mouse, you can say, so what? You're going to cure another mouse. We're actually putting it in mice that have been engrafted with human muscle. So now it's an animal. So it's a different type of animal model, a harder animal muscle model. But now it's a human now, because in the end, you still want to cure people, right? I mean, mice, we got enough of them. All right, so so this is basically the way I look at the therapies. We have, whoops. All right, I'm just about done. These are all the things, we got CRISPR, myostatin inhibition, we got morpholinos coming. A lot of labs, a dozen labs are working on this. MicroRNAs, anti-inflammatory, small molecules targeting epigenetic repression. I mean, basically, we're hitting around the world. I'm just one lab. Around the world, there's 40, maybe 70 labs, hitting all of these pathways, all with the goal. We don't need them all to work. We need one of them to work, and that's good news, right? There's a lot of things that we've learned. And I would say that this is incomplete because there's a lot we don't know, which is why we got to keep funding the basic reason. The more we know about disease mechanism, the more targets, the more therapeutic approaches we're going to have. Okay, because it's too important to get this wrong, right? We got to know as much as we can. And evidently the volume's not on. If the volume was on, you would hear Kelly O'Hara singing. What the heck is she singing? Oh, Finally See the Light. Seriously? It was on just a second ago. Anyway, she's singing We Finally See the Light, because I believe, and because we're kind of finishing this up, that's actually where we've gone through the basic discovery. We're now into therapeutic development, and I swear to God, I see the light. Therapeutically, I do. I honestly think that we're really getting there. So here's just some acknowledgments. I'd like to thank Mick for funding FSH research at UNR. FSH Society, there are a number of foundations that provide funding for us, and this is my group of geniuses that do all the work.